Welcome everybody to another exciting discussion at the SACS Forum. I have the pleasure today to be together with a friend and colleague who I've worked with for many years, Narender. And I think I'll first let him introduce himself, and then I'll introduce myself quickly.
Thank you, Florian. It's a pleasure to be able to talk with you through the forum of all the people. Let me just quickly introduce myself. My name is Narender Mantena. I have done various roles in the company. Most recently, and then specific to COVID, let me start with maybe a brief introduction of the company. So I work for a company called Biologically Limited. We are one of the oldest and largest vaccine manufacturing companies out of India. Just to give you a context, in 2020, we manufactured and sold over 700 million doses of vaccine. I've been with the company for nearly 30 years. As I said, donning various roles from business development, R&D strategy, and most recently, I look after our injectable business in US and Europe, as well as some of the partnerships and some of the strategies for the COVID vaccines.
Thank you, Narendra, for the introduction. Just before we go any further, I'll say just a few words about myself. I'm a physician and a microbiologist by training. I've been in vaccine research and development for almost 4 decades now, starting with academic research and then moving into industry, where for the longest time, about 15 years, I worked for Merck and was co-responsible for developing and licensing a number of vaccines and running the clinical trials to prove that they work. And after I left Merck, now roughly 11 years ago, I started a consulting business, and I provide research and development advice to a number of companies, and among them, just as examples, Moderna with their mRNA vaccines, and I have the honor and privilege to also sit on the scientific advisory board of Biologically E. And this goes back to a collaboration we already initiated while I was still at Merck, and several of my colleagues started to work with Biologically E at the time on a pneumococcal conjugate vaccine. I think we'll say a few words about that as well. And then when I left Merck, Narender and Mahima were kind enough to invite me to sit on their scientific advisory board. And I've since seen really and learned a lot from Biologically E and the collaboration with them, the transformation of a company that not only produces a huge volume of vaccines, but also has a long history of classical virology, vaccine development, and in general development expertise. And I want to mention this particularly here because in our haste to develop new things, we sometimes lose sight of the classical technologies. And we had, for example, I started my career as a student in the Bavarian Vaccine Institute, which was an institute that was founded relatively shortly after Jenner discovered vaccination or variolation at the time to provide smallpox vaccine. And it has actually been instrumental in bringing MVA to the public and introducing it to a very large number of people. And these kinds of institutes existed all over the globe. And of course, one of them is the one that Narender works for, that still exists. In many other places, these institutes were actually eventually dismantled and don't exist anymore. And that's a problem because we do not have the depth of classical knowledge anymore in many institutions that now develop vaccines. The corporations that do it at a large scale are relatively few in the meantime. There's been a huge concentration. And so I think there is an opportunity for cross-fertilization between those few institutions that have kept the classical knowledge and have built modern teams to develop up-to-date research and really modern approaches to vaccination like Biologically E. And so I mention all this here because I think it is important to understand where biological E comes from and where they have gone. And I'm very impressed by their team of scientists and developers that they've put together that has enabled them in record speed, very similar to the institutions in the US and Europe to develop, for example, a candidate COVID vaccine that's now in clinical testing, but also to develop already a second generation of pneumococcal conjugate vaccines, which are also in clinical testing. So a lot of progress has been made. And I think in talking about this here, I want to sort of go through the journey with you, Narender, and not just talk about the COVID vaccine, but sort of what made this possible and where you see things going. But over to you.
Thanks, Florian. I think, as you said, it's been a wonderful journey that we started, and I think that we hope to continue this journey to be able to make a difference, in the global community. So let me start with perhaps the two phases of biologically, especially the vaccine development. As I mentioned, we started vaccine development in biologically through a collaboration of through company called Evans Vaccines, which still the manufacturing facility still exists. So hence the name Biological E. At one time, it was called Biological Evans Limited, and this was this vaccine journey started in the early '60s by way of developing the bacterial vaccines for tetanus and later on diphtheria and pertussis. For most of so I would say '60s, '70s, and '80s, even into the '90s, we largely stuck to that group of vaccines, which is the tetanus toxoid and the TTP vaccine. Uh, there were a couple of reasons for it. One, vaccination as such was not a very lucrative business. You didn't have the public uh, awareness of the importance of vaccines, and even in India, it was a relatively, you didn't have a national immunization program. So and fortunately, and I wouldn't know the reasons why we kept those businesses at that time. Fortunately, we decided as a family to stay in the vaccine business while we grew the pharma business, especially with the growth of the Indian population and the Indian middle class. You saw the wave of Indian companies making generics in the '80s and '90s. While we weren't in the forefront of that, we did participate in that, but we kept the vaccine business even during those times, even though it was a very small contributor. I think that history of vaccine making really helped us when we decided to invest in the vaccine business, which was in the late '90s. And one of the reasons that we were able to do that was at that point the equity stake that Evans held eventually got transferred to Glaxo, and the family in the middle '90s towards the late '90s decided to buy out our equity partners, both Glaxo as well as Solvay, who we had a animal vaccine joint venture with. So by the late '90s, I think that the company was fully owned by the family, and at that point, when we decided to look at what we wanted to invest in to further our growth, the legacy of vaccines and the fact that, as compared to pharma and generic pharmaceuticals, the family felt that we can make a difference from contributing the globe as well as having a robust business plan remained in vaccines. Uh, I suppose, fortunately, during that time, there were a lot of initiatives in trying to make vaccines accessible to the global community, especially to the low income and lower middle income, with the various initiatives of early stages of Gavi, the Gates Foundation, the Wellcome Trust, having initiatives in making these vaccines available. So we started working in that ecosystem, and largely focused on our ability to manufacture and increasing the quality to qualify for WHO pre-qualification. In those early places, we really didn't do vaccine development. We were more, interested in partnering with companies, and we partnered with the largely academic institutes such as the Netherlands Vaccine Institute, as well as the Indian Institute of Science in India, and they gave the basic clones, which were I think generic vaccines for hepatitis B and Hib. And that's what allowed us to slowly develop the capability of process development, early stage process development, and some remnants of clinical development. It wasn't novel clinical development, but it was trying to do non-inferiority with the established correlates. So the most of the 2000s really laid the foundation for our process development and early stages of clinical development. I think fortunately, those investments and the risk that our family took in making those investments paid off such that by the end of the decade and throughout the next decade, which is from 2010 to 2020, we became one of the largest suppliers of those vaccines globally. And I think that obviously there's always a pressure on prices to make it affordable for the poorest countries. I think we were able to make these vaccines at reasonably affordable prices without compromising the quality. And I think that one of the inflection points during that discussions was our short-lived collaboration with Merck. I think that two things. It allowed us to start working with companies like Merck and understanding the quality requirements for global development. I think that laid the foundation for the next phase of our growth. I think probably most importantly allowed us to meet people like you and Barry Buckland and others that continue to be partner with us in traversing the next journey of our company, which is try to become a full-fledged vaccine development company. And the next phase was, leveraging those capabilities. We started looking at building a portfolio of vaccines that would allow us to start uh, expanding our technical portfolio from basic microbial vaccines to viral vaccines, as well as emerging technologies, just to broaden the portfolio of vaccines. So in that respect, I think the last decade also has been as transformational as the previous decade. We licensed number of vaccines, and I think we have about 8 vaccines that are pre-qualified by WHO, ranging from, as I mentioned, the diphtheria, tetanus, pertussis, the hepatitis B vaccine, the Hib vaccine. We have also added our Japanese encephalitis vaccine, measles and measles rubella vaccines in our portfolio and the various combinations of that. I think that now all of this worked, and currently we've expanded this portfolio to what Florian mentioned earlier. We leveraged our capability trying to develop a next generation vaccines in a couple of areas. One was the typhoid conjugate vaccine, and we're trying to develop a bivalent conjugate vaccine to address the paratyphoid. And I think most exciting part would be 2 programs we have internally. One is a pneumococcal conjugate vaccine, which is in phase 3, which is a 14-valent vaccine, as well as a 23-valent conjugate vaccine, which is going into phase 1 in about 3 months from now. I think that we're excited by this, that we'll not only have a vaccine for the developing world, we hope to be able to develop this vaccine for US and European markets. I think that largely reflects the path that what we have taken, to where we are right now. In addition to that, we have a IPV vaccine that we are developing that we can manufacture in less stringent containment guidelines, and hopefully that will be successful such that we can offer an affordable IPV vaccine to continue the elimination of polio. I think that leads us to where we are with COVID, and I'll take a pause right now for Florian to let me know which area should I go focus on now.
to let you breathe for a moment. Thank you, Narender, for this very nice overview. I think what you see here is that the persistence of a family and the continuous investment has led to a rather unique, fully integrated vaccine business, which is a rarity nowadays, and there are really just a handful of them around across the globe, where all the aspects of development, discovery, process development, and large-scale manufacturing at reasonable prices and high quality are combined, which of course makes this very, very exciting for people like me and Barry Buckland and Gerd Zettelmeisel, who sit on the scientific advisory board. And it allows us to essentially combine our interests in preventative medicine and in vaccine development with a reach into the whole gamut of development that kindly enough has been developed at Biological E and is in one hand. So that's really a great opportunity. And I think it'll be a successful business model, quite a different one from the one that has been developed under the different conditions of the markets in the pharmaceutical corporations in Northern America and Europe. But one that I think is complementary and that offers something that they may not have anymore, the complete integration and the experience to reduce it to a low cost, high volume, high quality practice, which is really, really interesting. And I think that leads us to COVID, where, as has been mentioned in a previous discussion, there is an unprecedented need for speed and quantity. And so what we have right now in the US, for example, we have now 3 licensed products, of which the first 2 both are mRNA based and very highly efficacious, have delivered about 50 or 60 million doses that have been vaccinated. But the global demand for a COVID vaccine at this point in time is estimated by CEPI to be about 14 billion doses. That is unprecedented for a single vaccine. Now, there are vaccines that globally, if you take them all together, produce, of course, more than 14 billion doses. But there is not a single vaccine that in this short period of time, first of all, has been developed, and secondly, has a demand for about 14 billion doses. And that brings me to where Biological E's efforts are at making a COVID vaccine candidate. And Narender, maybe you can explain a little bit where you stand.
Thanks, Florian. I think, as I said, I think perhaps, while appreciating the kind words that Florian said, let me talk about perhaps some of the lacuna that, biologically and other companies such as us faced, we're faced with when COVID broke out in about a year ago or about 13, 14 months ago. I think that as a company, while we were working on the next generation vaccines, such as the pneumococcal and the IPV, companies such as us really didn't have the basic research and discovery engine, historically. We relied upon technologies that were developed by other companies or other academic institutes, especially in the vaccine area. Because vaccines as such require such long-term basic investment before even a product comes out. I think that even, I think we were lucky that COVID, we were able to find the right target, but you can be argued that, it was bit of foresight in NIH that they supported some of the earlier coronavirus basic research, as well as the fact that spike protein worked. Need not have worked, and we would have been in a soup by then. But specifically to our company, when COVID broke, we really didn't have any existing research base that we could rely upon to discover our own antigen as such. While we knew the spike protein and the receptor binding domain were targets of choice from the previous coronavirus work, we really didn't have any a basis for us to quickly deploy our technology to develop a vaccine. At that time, we felt that the best way that we can contribute was to look for partners that can do the early stages and that had done some work. So we went and the way we approached it, we wanted to deliver what our capabilities were to be able to offer 2 kinds of solutions. One is to use a development engine to develop a product that we have large scale experience in, and also would be appropriate from a global community. And given the historical expertise we had in microbial fermentation, we targeted a product that was suitable for microbial fermentation, and it was logical the receptor binding domain made in yeast or E. coli would be a natural target for us. And I think that we were fortunate that we found a partner and partners that we were able to screen, quite a few potential candidates, and where we are right now is, I think that we partnered with a couple of academic institutes in the US, as well as the adjuvant suppliers all over the world, to be able to screen an appropriate recombinant protein vaccine targeting the receptor binding domain and adjuvanted to come up with what we think is could be an exciting product that will complement the existing licensed vaccines. So we were able to do all the preclinical work, and starting from about April, and we were able to have a product that was suitable enter clinic as early as September. And we are expecting the phase 1/2 data, where we're testing about 4 different formulations in the next 2 to 4 weeks. And based on the data, we are expecting to start a efficacy trial that will be run globally, along with an immunogenicity trial in India, starting as early as April. I think that these timelines are not unusual. Many companies have it. I think the most exciting part about about this particular product is our ability to commercialize it. I think that from a vaccine candidate perspective, we think that this will be a complement to the adenovirus vaccines as well as mRNA vaccines, because of the safety profile of how we delivered our hepatitis B and yeast programs. So this can be used either as a primary vaccination, potentially as a booster for both of the previous technologies. So I think that it offers a differentiated product profile to the one that are licensed right now. On top of that, I think it gives us the ability to leverage our capabilities in manufacturing. I think that if everything works out well, we'll be in a position to offer about 100 million doses per month, as starting from the second part of this year, and from July. So I think that by making this vaccine that leverages classical technologies, that leverages the very accepted yeast platform, delivering a product very similar to hepatitis B. I think that this kind of a product should make a difference and make a dent in the overall requirement of 14 billion doses that we need over the next couple of years. And I think we're fortunate that we were supported, in addition to the academic partners, as well as the adjuvant partners from the likes of Government of India, the Gates Foundation, and CEPI, to fund this development. In addition to that, I think that we looked at how we can also offer contract manufacturing services for other companies, and one of the publicly disclosed one is our partnership with Johnson and Johnson to manufacture their vaccine in India.
Narender, just that is a really good summary and very interesting. Just on the Johnson and Johnson collaboration, have you also made arrangements with them to test a prime boost regimen with their vaccine, one way or the other?
At this point, our relationship with Johnson and Johnson is primarily at the contract manufacturer. I think we are looking. I think we would explore heterologous boost of our vaccine with not only J&J but other programs also. I think we're just waiting for, hopefully, crossing our fingers, the data should come out in the next 2 to 4 weeks. Where I think, given the variants and given the nature of the disease, I think the heterologous boost seems to be a most appropriate opportunity for our product. And we'll not only look at Johnson and Johnson, we'll work with both CEPI and our country governments to test this concept amongst various primary vaccinations.
Yeah, thank you. I think that's what we're all hoping. The Johnson & Johnson product obviously has a favorable safety profile. It's shown to be efficacious, and it's just been authorized for emergency use authorization in the United States. And as I understand it, that product as well, at this point in time, is capacity constrained. And so it would be very, very valuable if you could produce larger quantities of the same product. But let me switch gears to another topic within COVID that we're all thinking about, and that is, of course, the various circulating variants and how good the vaccines will be. Now, the Johnson & Johnson product already has some data that suggests that it is quite protective against at least the South African variants and against one of the Brazilian variants, which is good news for all of us, I guess. Similarly, Pfizer and Moderna vaccines have shown to at least generate good neutralizing antibodies against these variants, so they probably will also work at a similar efficacy, one would hope. Where do you see the variants going, and what are you doing in preparation for them?
So I think that this was unforeseen and actually a foreseen problem, and that's the reason why I think the concept of the disease-induced herd immunity was not probably a good thing to talk about last year. I think that there was always a risk perceived by the scientific community about the variants, and unfortunately, the variants seem to be coming, and they seem to be causing trouble, especially people with prior disease or some vaccine immunity. So I think we have and we will be developing a vaccine towards the variant, and I think that given that we have not seen any efficacy data of the receptor binding domain, it'll be interesting to see whether the vaccines against the receptor type binding domain will have the same differences in the correlates or correlates such as the neutralization assay before we get some efficacy data. So we hope to have a variant in clinic either and I think that we're primarily targeting the variant that is out of South Africa. That seems to be a more difficult variant in terms of the efficacy with the existing vaccines. So we hope to have one in clinic as early as June. Now it's going to be a tricky part of how to have a licensure pathway for a company like us who at this point don't have efficacy data on the primary immunization. But I think that we have discussed with CEPI and a couple of country governments where we think that we will have a opportunity to do a placebo-based efficacy trial on the primary vaccination schedule and incorporate a variant as a booster or a heterologous boost, depending upon how the epidemiology shakes out over the next 6 months. So we think the fact that we can leverage this existing technology and do a variant probably is a very, very good opportunity that this can act as a booster towards certainly the adenovirus vector vaccines, where I think boostability has always been an issue, and potentially the mRNA vaccine, where we don't know the repeat vaccination, what it could do at this point. So we think a classically made protein vaccine, either targeting the spike protein or receptor binding domain with the variant, is going to be a necessary part of the arsenal to fight this disease later part of this year.
Yeah, I would agree, and I think that is actually potentially a very interesting complement to currently already existing vaccines. And in one scenario, I mean, even if your primary series is not yet licensed, you could imagine that a boosting part might actually get licensed earlier, because it will be important.
Yeah,
I think we're coming to the end, so unfortunately.
Yeah, I completely agree. I think that the concept of having a boosting vaccine without a primary vaccine not licensed is going to be an interesting challenge for us and the regulators as we go forward.
Okay, well, thank you again very much, Narender, and I think we'll conclude this. Thank you to everybody who has listened. Take good care.
Thank you, Florian. It's a pleasure once again.
It was a pleasure for me. Take care.
Bye-bye.